All right, chapter 24 continuing, beginning with slide 146. So we just finished the last of the accessory organs. We did the liver, the gallbladder, and the pancreas. And so now we're going to pick up with the small intestine. So the food will go out of the stomach through the pyloric sphincter and enter the first section of the small intestine. Now, the small intestine is one of the longest organs of your body. It actually runs about 23 feet long. And it's a long muscular tube where we're going to finish all of the rest of the chemical digestion. There is a little bit of churning, but it's not nearly as efficient as the mechanical digestion of the mouth or the stomach. So mainly we're going to concentrate on chemical digestion and absorption. 90% of nutrient absorption happens in the small intestine. And so we divide the small intestine into 3 different regions, and what changes is actually the kind of setup of the tissue inside changes a little bit through each section. And the 3 sections in order are the duodenum, the jejunum, and the ileum. The duodenum is the smallest of the segments. It's right closest to the stomach, attached to the stomach, and it's pretty much just a little curve shape. It's only 25 centimeters long. It's the mixing bowl. It gets the stuff from the stomach, which is slightly acidic, and so it's going to balance that with the digestive enzymes and buffers from the pancreas and with the bile from the liver and the gallbladder. The jejunum is the middle one, and it's 2.5 meters long, and it's where most of the chemical digestion and nutrient absorption occur. So by this point, by the jejunum, we've added all the enzymes we're going to add. We're just letting the enzymes work and break down all of the food into the macromolecules, into their monomers. And then we absorb it across the intestinal wall. And finally, the ileum is the last section is the longest section. It ends at the joining of the ileum to the large intestine, to an area where we have a little cup in the large intestine called the cecum. And so what controls the movement of the food from the small intestine to the large intestine is the ileocecal valve. Notice this is the only valve on the entire intestine. We have the opening valve, the pyloric sphincter, and there are no valves throughout the jejunum or the duodenum. And only at the very end of the ileum do we have another sphincter. So it will remove the food from the small intestine and allow it to pass into the cecum part of the large intestine. So here's a diagram, and they kind of color-coded it for you. So only the first 25 centimeters are the duodenum. And then we get into the jejunum here in purple. And then we enter the ileum, which is the longest section. And then the ileum curves back around and joins the large intestine at the ileocecal valve. This is a cadaver ileocecal valve. Okay. So inside the small intestine, again, absorption is the goal, and absorption requires lots and lots and lots of surface area. So we have these circular folds that are inside the intestinal lining, and we've already seen these in the very beginning of this chapter. Now, unlike the folds of the stomach, these don't go anywhere. They stay folded all the time because, again, they are to hold more intestinal villi, which hold more intestinal microvilli, so we have the maximal surface area that we can use in order to absorb all of the nutrients. And in the lab, we don't call them circular folds. We call them plicae, P-L-I-C-A-E. So if you see the plicae on your list, it's referring to the same thing, the circular folds. So on the circular folds, we have intestinal villi. They're covered with simple columnar epithelium, which usually has microvilli that forms what we call the brush border. So here are the circular folds or plicae. This is looking at the jejunum. And then if we're looking at the histology of the small intestine, we're going to have kind of the same layers we had before: mucosa, submucosa, muscle layer, and serosa. We're going to add in a few more things. We're going to add in lacteal. So up in each villus, each little finger-like projection on the circular fold, we're going to add a special vessel that is there to absorb fat droplets. It's going to transport chylomicrons, which are fat spheres that are too large to enter the blood capillaries. So we take a lot of the digested fat directly through the lymphatic system. Now, why are they called lacteals instead of lymph something? Well, because all the added fat in the lymph makes it look cloudy and milky. So they named these vessels lacteals as they're holding this fat-laden lymph fluid. We're also going to add in intestinal glands. Now, we had gastric glands in the stomach, so this is kind of like the intestine's answer. These are called intestinal crypts. So they go deep into the lamina propria. At the bottom, we have stem cells because we're going to be replacing these often. And we're going to have Paneth cells that are going to be there for immunity. And we're also going to have our enteroendocrine cells, which are going to secrete all the different hormones we will discuss related to the intestine. So there's a segment of the small intestine. Again, you see the plicae or circular folds. And then if we were to look up close, we would see these villi on the circular folds, and in each villus we should have an artery and a vein and a lacteal. Or I guess technically there'd be like a little capillary loop here, right? Okay, and then between the villi, we have the little, where it goes down deeper, those are our intestinal glands, and we might have some lymphoid nodules in there. And then we'll have our muscularis mucosae, all that was part of the mucosal layer. Then we have our submucosa, our muscular layer, and our serosa. Now notice our submucosal plexus is here also, the nerves, this yellow. Okay, so we have arteries and veins and lacteals and nerves. Submucosal plexus in the submucosa, our myenteric plexus between the circular and longitudinal layers of the muscularis externa. And if we look at the very bottom of the intestinal crypt, this is where the Paneth cells are that are going to help secrete some of the enzymes that we're going to see. Now, this is how the lacteal stands up in a villus, and we have our capillary network around it. So we're bringing oxygen to all of these cells with this side, and we're absorbing all of the nutrients and putting them in the blood, and we're absorbing the fat into the lacteal. And here's a micrograph of a villus. Okay, so in the duodenum, we have submucosal glands. So they're going to be in the submucosa that are going to make lots and lots and lots of mucus. Now, what does mucus do? Just like the cells lining the stomach made mucus to combat the acid, the duodenum is going to be receiving the chyme from the stomach. And if any of that acid is still there, we need to protect the walls of the duodenum from acidic damage. It also has bicarbonate ions that are secreted, so start to neutralize the low pH and make it higher and higher and closer to neutral. Every day, your small intestine makes almost 2 liters of intestinal juice. Now, what is intestinal juice for? It's to moisten the chyme, make it even thinner and waterier, less pasty, assist in getting rid of the stomach acid and buffering the pH, and helps keep the digestive enzymes in solution along with the products of digestion, the little individual monomers. We want to keep them in solution until we absorb them. The mucosal layer also makes a few enzymes. The brush border makes the brush border enzymes, and they can break down materials that come in contact with the brush border. We also make enteropeptidase as a particular brush border enzyme, and you see peptidase, so you know that's going to have something to do with proteins. This activates pancreatic trypsinogen. So remember we said we take trypsinogen and cleave it? Well, this enzyme, enteropeptidase, cleaves the trypsin, and then the active trypsin cleaves all of the other protein enzymes. So the chyme comes into the duodenum through the pyloric sphincter, and the muscles in the duodenum have very weak peristaltic contractions that move it very slowly towards the jejunum. We want it to move slowly because, again, this is our last opportunity to introduce most of the digestive enzymes coming from the pancreas, plus the bile. The contractions are myenteric reflexes that are not controlled by the central nervous system, but parasympathetic stimulation will accelerate them. There's the gastroenteric reflex that causes motility all along the entire small intestine. The gastroileal reflex that triggers the opening of the ileocecal valve. and that's moving the materials from the small intestine to the large intestine. And it has the opposite effect from the enterogastric reflex, which closes the pyloric sphincter. Okay, so here's a little summary of the different reflexes. Neural and hormonal mechanisms will help control the digestive glands, and they're centered on the duodenum because that's, again, where the acids are neutralized and the enzymes are added. After that, we're pretty much just letting the enzymes work and absorbing the results. The nervous system control of the first section of the digestive tract is preparing it for activity by increasing parasympathetic stimulation. We also are going to inhibit gastrointestinal activity if there's sympathetic activation. And we're going to use reflexes to coordinate movement, and motor neuron synapses in the digestive tract release neurotransmitters. We also have our intestinal hormones. So we have our peptide hormones. Now these are different from the peptide enzymes. The peptide hormones are going to be controlling the movement through the digestive tract. We're going to cover several regions of the digestive tract and tell the accessory glandular organs what to do. So these hormones are gastrin, which we saw in the stomach, secretin, gastric inhibitory peptide, cholecystokinin, vasoactive intestinal peptide, and enterocrinin. Gastrin, again, comes from the stomach, but we also have G cells in the duodenum. So if it sees incompletely digested proteins, remember a lot of the protein digestion has to happen in the stomach. If those proteins enter the small intestine are still largely undigested, these G cells will secrete more gastrin, which tells the stomach to increase its motility. And it also makes us make more acids and enzymes. Secretin is released when the chyme gets to the duodenum, and it tells the duodenum to go ahead and release those buffers to raise the pH, and tells the pancreas and bile ducts to get ready. It reduces gastric motility because the stomach is emptying and the secretory rate of the stomach. Gastric inhibitory peptide is secreted when fats and carbs enter the small intestine because we're done with the gastric phase and we're starting the intestinal phase. And we've already discussed cholecystokinin, which is going to be looking for high amounts of fat. And it's going to open the hepatopancreatic sphincter and allow the bile to enter the duodenum. Vasoactive intestinal peptide stimulates the intestinal glands to go ahead and secrete all of their secretions, dilates the capillaries, and inhibits the stomach from producing additional acid. And enterocrinin released when the chyme enters the duodenum and stimulates additional alkaline mucus production to counteract the stomach acid. I know that was a long list of hormones. And here they are kind of summarized for you, where they act and what they do. And the red arrows mean they stimulate these things, and the blue, like dead ends, means it inhibits. Okay, so again, the name of the game is absorption. So we're using these chemical enzymes to break down our proteins into amino acids, our carbohydrates into simple sugars, our RNA and DNA into nucleotides, and what did I leave out? Proteins, amino acids, carbs, fatty acids of the lipids. And then we have to take these small monomers and absorb them over the surface of the mucosa and get them into the circulation. So we actually move the mucosa to make it even more absorptive by allowing the food to pass over it several times. Finally, we finish with the absorption of the small intestine, and what enters the large intestine is largely the indigestible waste products. Now, the large intestine is also called the large bowel or the colon, and it starts at the ileocecal junction and goes up the right side of the abdomen, and then it curves, goes around the top of the abdomen just under the diaphragm, curves again, goes down the left side of the body, curves backwards, and then will form the rectum, which leads to the anus. So it goes under the stomach and liver, frames around the small intestine, and it's only about 1.5 meters long, but it is very much wider than the small intestine. So parts of the large intestine. The first opening is actually below the level of where the small intestine enters. And so this is like a little bowl or cup shape called the cecum. Hanging off the bottom of the cecum, by the way, is a small elongated section called your vermiform appendix. Vermiform means worm-like. And the vermiform appendix is a way to hold on to your intestinal bacteria and give us a library of bacteria to replenish if you take antibiotics. The colon is the largest portion and it goes up and across and down. And then the rectum is only the last 15 centimeters leading to the elimination point or the anus. So the cecum, a expanded little cup or pouch, receives and stores materials coming from the small intestine, the ileum section. And it begins the process that's the focus of the large intestine, which is compaction. So we spent a lot of time adding fluid to the foodstuffs. So we went from a bolus to chyme, and then we added even more watery secretions to make it very loose. Now we want to do the opposite. We want to reabsorb as much of that water as we can to create a compacted material that can be pushed out by the small smooth muscles of the rectum and anus. Here's the information about the appendix. I kind of got ahead of my slide there. Now, the colon has a larger diameter and a thinner wall than the small intestine. And all along, there are going to be these little pouches called haustra, and the plural is haustrae. We put an E at the end, that allow the colon to expand sideways, or the diameter, and elongate. Holding these together are these bands or ribbons of smooth muscle that run down the center called the tenae coli. 3 longitudinal bands. They're deep to the serosa, and they're kind of like the outer layer of the muscular layer, and it help move things along. Muscle tone in the tenae coli creates the pouches, so it's kind of gathering up the colon. We also have a bunch of fat dropules that are attached called the omental appendices, and they're hanging on to the serosa of the colon. So again, the 4 regions of the colon, once we get past the cecum, we have the ascending colon on the right, the transverse colon crossing over the body, the descending colon down the left side, then it turns and goes backwards. It has an S shape called the sigmoid colon. The ascending colon begins at the top of the cecum. And when it gets up to the top near the liver, we have the right colic flexure, also known as the hepatic flexure. Then we enter the transverse colon. Goes across the abdomen from the right side to the left side. When it reaches the left side, which is right where the spleen is, it has the left colic flexure, which turns it downward, also known as the splenic flexure. And remember, these are held in place by the mesocolon. And then it begins the descending colon, goes down the left side to the iliac fossa. And all of this section is retroperitoneal, which means it's not contained within the peritoneal cavity. Instead, it is behind and is attached to the abdominal wall. Last 15 segments is an S shape that is behind the urinary bladder, suspended from the sigmoid mesocolon, and it empties into one final storage area called the rectum. So what's the job of the large intestine? Well, first of all, again, it's for compaction. It's for removal of water. It is also a large place for bacterial colonies to break down the parts of your food that you couldn't. And we actually depend on these bacterial colonies in our colon to give us some of our vitamins and minerals that we cannot absorb on our own. So, since it is still going to be doing a little bit of absorption of nutrients, we have a large blood supply that comes from the superior mesenteric and inferior mesenteric arteries, and the venous blood is collected by superior mesenteric and inferior mesenteric veins. Whew, there's our diagram. Okay, so here's the ileum, the last part of the small intestine. Here's the ileocecal valve. Here is the cecum. And then here is our vermiform appendix. Now we begin the ascending colon, transverse colon, descending colon, sigmoid colon, and rectum. Now, these bands here are the tenae coli that are forming the gatherings known as the haustrae. And here are our mental appendices, these little fat droplets lining the tenae coli. The last section is known as the rectum. It's only the last 15 centimeters, and it is an expandable organ to temporarily store feces. Feces is what we create after we have compacted and dehydrated the food waste. The anal canal starts at the last section of the rectum, and it has small longitudinal folds that are called the anal columns. And then finally, the opening leaving the body is known as the anus. It is the exit of the anal canal, and it has keratinized epidermal cells like your skin on the rest of your body. Now, you might not know this, there are actually two anal sphincters. There's an internal sphincter and an external sphincter. The internal sphincter is involuntary muscle. The external sphincter is voluntary. The internal one is circular muscle layer, smooth muscle cells. The external one is skeletal muscle fibers, and again, voluntary control. So here is the rectum, last 15 centimeters. These are the anal columns. Here is the anal canal. This curve right here is the internal anal sphincter, and right here would be the external anal sphincter muscle. So this ring of muscle is internal, this ring of muscle is external. The large intestine doesn't have villi because it does not need to do as much absorption as the small intestine does. So we don't have to worry about surface area. It does have an abundance of goblet cells to make sure that it keeps moving things along by producing mucus. And it has its own kinds of intestinal glands. They're deeper than the glands of the small intestine, and they're mostly, again, goblet cells for mucus production. That gives us lubrication for the fecal material to keep moving along. We also have large lymphoid nodules throughout the lamina propria and submucosa because, again, we want our helpful bacteria to colonize. We want to keep the bad bacteria to a minimum. So we need to make sure we have areas that are monitoring the bacterial colonies to figure out if they're helpful or harmful. So looking at a close-up of the wall of one of these house stray. Simple columnar epithelium at the surface facing the lumen. Goblet cells down in the intestinal glands. But we still have basically the same layers. And here is a photomicrograph. Functions of the large intestine: to reabsorb or absorb water, a little bit of nutrients, less than 10%, bile salts, we can get them back, we would like to recycle them, organic wastes, vitamins and toxins produced by bacteria, to compact all of the contents into feces and to store it until there's enough material to warrant the process of defecation. So as I mentioned, it's a large home for many, many, many helpful bacteria. So the bacteria that live in your colon are known as your microbiome. And there could be several different kinds of microbes. Bacteria, fungi, and viruses make up your entire body's microbiome. But in your colon in particular, we really want it just to be these particular kinds of bacteria. Vitamins or organic molecules that are needed in chemical reactions as cofactors or coenzymes. And normal, helpful bacteria that live in your colon make 3 vitamins that you cannot make and cannot absorb from your food. So these bacteria are required for these 3 vitamins. Those vitamins are vitamin K. Vitamin K, you might remember, is needed for blood clotting. The second vitamin is biotin, which is important in glucose metabolism. And the third one is vitamin B5, which is pantothenic acid. Now, vitamin K is fat soluble. Biotin and vitamin B5 are water soluble. We need vitamin B5 in order to make our steroid hormones and some of our neurotransmitters. All right, the organic wastes. We talked a little bit about this when we talked about hemoglobin breakdown. So the bacteria convert bilirubin to urobilinogens and stercobilinogens. And some of these go into the bloodstream and go into urine. They're the ones that make your urine yellow. The urobilinogens and stercobilinogens are converted. Notice they had ogen at the end. So they get converted in the colon to urobilins and stercobilins when they're exposed to oxygen. And they are what make your poop consistently brown. These are just breakdown products of hemoglobin, the red pigment in your blood that binds oxygen. Bacteria break down any peptides that are left over in feces and they generate ammonia. Now, they generate it as a soluble ammonium ion. They also create some compounds that are not so pleasant. Indole and skatole are nitrogen-containing compounds that make your poop smell bad, along with hydrogen sulfide gas that adds that rotten egg smell from the sulfur. Bacteria feed on your indigestible carbohydrates, and they produce gas in the process. They produce flatus, which is your intestinal gas in the large intestine. Unfortunately, this produces farts. Air that you swallow during eating produces burps. If it is not released vertically as a burp, it will also pass through the colon and be added to the flatus of the bacteria. Isn't that fun? We're talking about farts today. The motility of the large intestine. So we have reflexes that we had in the small intestine affect the large intestine. The gastroileal and gastroenteric reflexes will move the materials from the small intestine to the cecum of the large intestine while you eat. It increases the speed of moving things through your large intestine. So when you eat a new meal, your body begins to speed up getting rid of your last meal. The movement from the cecum up to the transverse colon, however, is very slow because number one, we're fighting gravity. Number two, we're giving the body extra time to absorb as much water as we can. We use the same kind of peristalsis to move the material along the length, and we even use segmentation to mix the contents of the haustra with some of the added things that are like the bacteria are adding. Movement from the transverse colon through the rest of the large intestine results from powerful peristalsis. These are sometimes called mass movements. The stimulus is when the stomach gets stretched and the duodenum gets stretched. So that lets your body know that more food is on its way into the intestines. That the distension of the rectal wall initiates the defecation reflex. We have two positive feedback loops, and both of them are triggered by the rectum being stretched by stretch receptors. One of them is the intrinsic myenteric defecation reflex, and it's going to cause contractions of the sigmoid colon and the rectum. And then there's the parasympathetic defecation reflex, which is longer, and it stimulates mass movements. Okay, to eliminate feces, we must relax both internal and external anal sphincters. The reflexes control the internal one. Voluntary control controls the external one. And it's carried through the pudendal nerves. And here's a lovely graphic summarizing this process. All right, so I'm going to stop this one here. The last PowerPoint will only be slides 201 to 219.